Hello, everyone, and welcome to today's AAPS Chalk Talk. Today's Chalk Talk is hosted by Mercodia. You may submit your questions at any time by entering them into the Q&A box of the Zoom platform. All questions will be answered at the end of the program. It is now my pleasure to introduce today's moderator, Emma Ranka. Emma, the floor is yours.
Thank you very much, Stacy and AAPS. Hello, everyone, and thank you for joining us today. My name is Emma Ranka, and I'll be moderating the Q&A session of today's webinar. We have a very exciting presentation ahead of us. It's my pleasure to introduce Sibel Amedova, who is a project manager at Mercodia. With hands-on experience in data handling and data transfer agreements, Sibel brings valuable insights into the complexities and best practices of DTAs. Today, she will guide us through the essentials of data transfer agreements, covering key points and common challenges. Sibel, the screen is yours.
Thank you, Emma. First, I would like to thank AAPS for the opportunity and providing this platform. Today, I will be talking about building a comprehensive data transfer agreement, best practices, and hands-on experience. In this webinar, I will be going through the importance of data transfer agreements in bioanalysis by focusing on data security, compliance, and key roles in multi-site trials. I will be also talking about some practical strategies with a case study to develop an effective DTA for seamless data transfer. In the end of this session, we will have a better understanding the key components of DTAs. I will also talk about common data transfer challenges and developing skills to identify and resolve issues more proactively during the DTA negotiations. But before I go through this session, I would like to first introduce myself a little bit. I work as a project manager within the bioanalysis and services group in Mercodia. I have also worked with the LIMS system deeply, both as an end user and also as a system administrator. So I have an experience managing and handling data accurately. I'm also responsible for evaluating quotes, agreements, and managing customer interactions. And I hold a master's degree in biotechnology from Uppsala University in Sweden. First, I would like to start with explaining what is a DTA. A data transfer agreement is a document that outlines how data is transferred, used, and protected between parties in a research study. So it is basically a document that helps parties to transfer the produced data in a negotiated or agreed way in a research study. And why is a DTA critical? High quality data allows researchers to make well-supported conclusions and advancements. Accurate data is essential for study results to be replicated and validated. DTA helps protecting the sensitivity of the bioanalytical data. And sensitive data in bioanalytical studies includes like personal health information. So it must be protected to prevent privacy issues. And laws like GDPR also require this protection to maintain trust and confidentiality. Keeping data secure provides accurate research results and protects valuable findings from misuse. It also ensures that data is following the regulatory compliance within the scope of the study. And it enhances the collaboration and gives a smooth way of handling the study. And to start with efficient DTA, we need to understand the key components of it. To start, let's define what we mean by bioanalytical data in this context. This includes raw data, process data, or method validation reports. Setting these definitions help clarify the scope of data and management of it. Regulatory compliance is also critical. We follow GXP guidelines and meet expectations from authorities like FDA, EMA. This ensures that our study adheres to global standards, which is essential for data credibility and regulatory acceptance. And next, we have to specify what type of bioanalytical data to be transferred. Is it a raw data? Is it a process data? And we also outline the required formats, like if it's a CSV file, or does it need a password protection? So this ensures everybody knows exactly what to expect in data exchange. And protecting data privacy is highly important. We comply with industry standards and ensure confidentiality and security participant information throughout the study. In terms of roles and responsibilities, we clearly outline the duties of each party involved, like sponsor, CRO, and test sites. And this way, everyone knows their obligations and can work together efficiently. And finally, we define the duration and conditions for ending the agreement. This provides clear expectations on when and how the agreement may terminate, and ensuring transparency between all parties. So this structured approach to data management helps us maintain accuracy, security, regulatory compliances, and ultimately supporting a successful study. And here I would like to go through a bit more roles and responsibilities in a bioanalytical DTA within a data flow overview. In a bioanalytical study, first we need to have biological samples, right? And these are usually collected in collection sites or clinical research units, it can be called. And they provide the initial data, for example, a sample manifest. And this sample manifest can include the subject IDs, collection time, collection points, visit days. Once all these initial data is ready, samples are transferred together with sample manifest to test sites to process and analyze them and generate the data. Once the results are ready, they need to be transferred to the CRO within the standards of the study. And CRO responsible with managing the data flow between all these labs and also the sponsor, and ensure the quality and regulatory compliances of the study. And sponsor is the part that oversees the entire study and ensuring the data transferred meets regulatory and study-specific requirements. And of course, there are some challenges during this whole process. Data privacy concerns is one of them. So ensuring secure transfer and storage of sensitive patient data while complying with all these regulations is really important. And adhering to strict regulatory frameworks might be challenging as they might vary across the regions. Cross-functional coordination is also the biggest challenge, I would say. Different stakeholders have conflicting expectations about the data delivery, and these incorrect assumptions about how the process can cause miscommunication and might end up with delays in the study. Data quality and integrity is another concern during these studies. Ensuring the bioanalytical data accuracy and consistency across multiple sites and stakeholders is very challenging. Data ownership and IP protection can be a concern, and managing all these intellectual property rights and clarifying data ownership in the context of the methods. And the timelines and data availability. For example, once a clinical research unit has a procrastination or postponing a patient recruitment, it might delay the sample transfer to the test site, and that can also result delays in the sample analysis and data availability, which can lead to delay in the studies. So here I have a case study that I would like to present and have a deeper look into the real world example. So we have a recent clinical trial for a new drug, and the pharmaceutical company faced some challenges in ensuring consistent data transfer and quality across multiple sites during phase 1. So in this case, sponsor is the pharmaceutical company, and they are collaborating with CRO. And CRO is the main communication point between the sponsor and all the labs that they will perform the sample analysis. To have a little bit of more clear understanding, I have played some roles here that is involved during the study. So clinical lab manager collection sites is collecting the samples and preparing the sample manifest. And they need to send the samples to the test site. And some of the samples and methods is not available in the test site, so they have subcontractors to do the measurement with them. And that means a data transfer agreement needs to be made between all these parties for a smooth transition. Once a sample manifest is prepared, the test site takes this and prepares blinded and unblinded data for both managers that is working in CRO. And once the samples are measured and analyzed, all these data is QC controlled and transferred to the data managers, to the both blinded and unblinded. And since this is a multi-site study, they faced some challenges. Diverse system requirements. Different sites were using varied bioanalytical methods and systems and data formats, and that was leading to inconsistencies in data. And this fragmentation is also causing the delay in sample analysis and data delivery. And of course, it's also raising the concerns about the data integrity. During the study, there were also some corrections needed to be done, mainly in the blinded data. This was mainly the correction of the collection time points. And this blinded data is generated from the information in the sample manifest, as it contains randomization IDs and collection times and etc. So initial corrections needed to be done in collection site, but not in the test site. And unblinded data contains both information from sample manifest and the measured result. So several issues arose here from the stakeholders, but by not fully understanding where the data is originated. As I mentioned in this slide, if a collection time needed to be corrected, it needs to come from the clinical lab manager, as this data is provided by them, but not the test site manager. While blinded and unblinded data managers asked corrected to the test site, as they were not aware where this data was originated from. So these knowledge gaps ended up having reactive management. Often stakeholders were not prepared to address the issues proactively, and they had misaligned expectations. In order to solve this, we have harmonized the path. We have standardized our DTA development. We have collaborated with all stakeholders to establish the protocols, including sample labeling and uniform data formats for all the data that is produced. We have also provided comprehensive training for everyone, focusing on the data sources and the workflows. We have developed a clear workflow for handling data by clarifying the journey of it for both blinded and unblinded. And all those things resulted with creating a feedback mechanism where it allows people to address misunderstanding and continuous improvement. So the outcome was from this study was improved data integrity. All these harmonization efforts significantly improved the data and quality and consistency across all sites. Stakeholders were engaged more proactively. They had a better understanding of the data flows, and that has reduced delays and errors during the study. This also enhanced our collaboration between multiple sites, clearing the roles and responsibilities facilitators with better communication among all sponsors, CRO, and test sites. And thank you for all listening. If you have any questions, I'm happy to take it.
Thank you, Sibel, for a very interesting presentation. So now it is time for the Q&A session. And as we said in the beginning, you can submit them in the Q&A box, and we will read them and ask Sibel. So let's see, we think we have a question here. Sibel, who is responsible for defining stakeholders related to DTA, and who is responsible for coordinating the DTA?
This can vary based on how your study is built up, but I can give an example already based on this story. So this was a case that we have experienced, and we were a test site here, and we have initiated the DTA collaboration and communication mainly. But I believe in this case, it will be better if CRO initiated all those things, communications, as they are the main contact between all these sites. So they are aware of the expectations way better than a branch of a study.
Okay, thank you. I can see another question here. It seems like the DTA is a complex process. how long time does it usually take to finalize a DTA?
This can also vary depending on how your study built and also how engaged stakeholders you have. In a complex study like this, it might take up to 2 months because a lot of people are involved and a lot of people are reviewing the process. But it can also take even longer time if you had more sites that are involved. And how your data is generated, what are the negotiations? Because I mentioned all systems are different and the outcome of the systems are also different. So it takes time to adjust everything a little bit and harmonize it.
I see, thank you. Another question here is, what is the biggest challenge you have faced when working with DTAs?
Biggest challenge usually comes from the knowledge gaps. When I work with the CRO, often blinded data managers and unblinded data managers are introduced study to the very later stage, and they don't really have the concept of the clinical lab collection site, and they don't have the really concept of the data flow. So it is a really hard to explain how things are coming to our plate and how we are generating data here. And once there is a mistake happen in collection site, it is really hard for us to manage that part of and provide the all communication flow. I think that is the biggest challenge I usually face almost in every study.
Okay, thank you. I have another question here. How does the DTA comply to GDPR or other privacy regulations? For example, PIA or TIA. I know you talked a little bit about GDPR.
Yeah, this is usually how you secure your data, where you transfer your data, what platforms you're using, and what is the scope of your study. If it's a PK study, of course, things are a little bit more stricted, and if it's an exploratory study, it can be a little bit loose. But usually, we are using randomization IDs during the study that we don't have the exact information of the patients. And once we transfer, we are creating secure platforms that it doesn't allow the leak of the data other than the people who has access for it. And we are also using password protection, and only relevant blinded data manager receives this password to encrypt all data, basically.
Okay, perfect. I can see we have another question here. Can you please discuss a little more about the difference between blinded and unblinded data?
Yeah, I had actually a little bit of an example that I have prepared in case this question comes up. Maybe I can go through that. This is a dummy data that I have created today, and it is just an example of how we do in the studies. This is the blind data format that you can see. You just have a randomization number of your subjects and your protocol IDs and your study periods, if you have a period in your study, and which days and time your samples are collected for what analyte, right? And this data comes from the sample manifest, and blinded data helps usually the data managers to follow if they are studies incomplete, and they can also prepare in advance their platforms for the measured data when it comes. And unblinded data is containing the real measured concentrations of, or whatever method you're using, calculated concentrations of your samples, basically. And there you can do your statistical analysis based on your patients, how they behave with the drug that you test on them.
Yes, thank you. We have another question. In the case study, you standardized the DTA development. How would you recommend us to improve collaboration with stakeholders?
I would say in those scenarios, it is better to gather all stakeholders together for a DTA session and ask what their expectations are and mention the limitations of your systems. So start negotiating actively in live what can be given and what can be handled by which system, right? And in my case, I have tried many times doing this by email, and it usually takes a very long time. But once we have a live meeting, it fixes 95% of your problems, and people align very quickly. Yeah.
Thank you. Okay, let me see. I have another question here. What would you recommend if collection sites frequently deviate from uniform or agreed upon data formatting? What actions would you recommend a CRO to take to standardize the lab data?
First, I would recommend to CRO to bring test sites and these collection sites together and let them discuss a little bit, because every collection site has their own way of working. Some are using barcodes to keep track of their system, some work a little bit more manual, but more or less most of the collection sites can have an Excel file outcome from their system, right? And this is also a concern of test sites, not just CROs, if they can handle the outcome of the sample manifest. So I would prefer to bring them together and ask what data formats your company can handle with.
Good, thank you. Yes, I think that was all the questions that I got here in the Q&A box. But please feel free to email Sibel after this webinar. You have our email address here, and you can also contact Marcodia through our info email. Yes. Thank you very much for listening.